develop into? After the fertilization, the zygote develop into embryo. Then pen develop into pen. Primary endosperm nucleus develop into who will speak? The primary endosperm nucleus develop into hello. What would be answered? In our last class, already you had written your primary endosperm nucleus develop into endosperm. Then your ovule develop into hello. Ovule develop into speak. After the
seed,
the ovule develop into
seed,
seed.
seed.
Then integument develop into seed coat. The outer integument develop into the outer integument develop into the outer integument develop into. Then what is the outermost layer of seed coat? What is the outermost layer of the seed coat? Or simply on then outer seed coat is known as dash. Someone can answer? The outer seed coat known as the testa and the inner known as the tegmen. The testa is developed from the outer integument, whereas the tegmen developed from the inner integument. Now tell me what develop into our the fruit after fertilization? What develop into fruit? The ovary develop into fruit. Then, and what is the primary function of endosperm? What is the primary function of endosperm? What is the primary?
It nourishes the
To nourish the zygote which develop into embryo. And then, and you might have seen that, and in case of the groundnut seed, then and when we separate the two cotyledon, then there is a small space there. Then there at the middle of the seed, then there is a small depression is there. Have you and then have you ever noticed? Have you ever repeated? Then reported? And when we break open the groundnut seed, then we have the two cotyledon. Then when we closely observe this cotyledon, then we'll find then there is a straight portion is there. Then there is a straight and a depression portion is in the cotyledon. Then have you reported? Then have you ever noticed? Yes or no? Hello. अरे बादाम जो भांगी सारा मझी देखिए गोटे छोटिया गार पड़ी दी फल देखिच से जगह आगुरी किए थिला हमर एंडोस्पर्म थिला ताको एम्ब्रियो खाई देछि हैला सेति सेटा फांका हो जाइछि द सीड हैविंग एंडोस्पर्म नोन एज एंडोस्पर्मिक सीड especially endospermic seed found in our monocot grasses rice maize etc whereas the seed do not have endosperm known as a non endospermic seed or X albuminous seed, especially dicot seeds are X albuminous seeds because they do not have any endosperm. Got this point? Hello. Hello.
Yes, sir.
Okay. So that now here and we have discussed about now today the development of embryo. Then actually now tell me what's your idea about embryo? What's your idea about embryo?
Fertilized egg or zygote develop all round.
Then what's the concept of embryo? Actually,
Fertilized egg or zygote develops a resisting wall around
no,
itself
no,
and
no, no,
forms
no,
oospot and
no, no.
embryo.
Then it is not an embryo. Embryo means what? First the egg and a first your male gamete and female gamete combined to form zygote. Zygote in course of development to form embryo. You may speak then in the plant. You may speak the small miniature or immature plant that found inside the seed is known as an embryo. Got this point? ମଞ୍ଜି ଭିତରେ ଥିବା ଛୁଆ ଗଛଟାକୁ ଆମେ embryo ବୋଲି କହୁ. And then you all have seen the embryo. And for example, you take same example of the groundnut seed नहीं जाओ seed टाको भी भांगी दियो. देखो तार गोटे साइड रे देखिबो से जो मंजी जो दी फळा हे जीवो धळा धळा गोटे साइड देखि गोटे छोटिया गोटे लागि जे गोटे छोटिया गोटे टिकेटा गोटे लागि कि अछि. Then have you ever reported this? Hello. Yes or no?
Yes, sir.
Yes, sir. Yes, sir.
And then this is the embryo proper, and the two cotyledon that you are eating, the two white portion that you are eating, they are also part of embryo. But the proper embryo or the embryo proper store the food material in the cotyledon. The two part you are eating, these are the part of embryo, but embryo store the food material in them. They are the storehouse of the embryo. Then on the cotyledon. And then the process of formation of embryo from the zygote is known as embryogenesis. The process by which embryo are formed from the zygote, but the course of development is known as the embryogenesis. And then, so that and first we have to look about, and before we discuss about the embryogenesis in details, so first we discuss about the structure of the embryo. Actually, then what are the part of the embryos? Look at this figure. Then this is look at the extreme right. Then this is our embryo. Then a typical embryo having, look at here, this is the hypocotyl is there, cotyledon is there, epicotyl is there, as well as the plumule is there. Okay. And here you are seeing the foliage leaf. In place of foliage leaf, actually this is the advanced stage. But in case of the earlier stage, here we are having what? The small leaves. The young leaves are present here, or the leaf primary are present here. This complete is known as the plumule. Okay, so that a typical embryo having radicle. Then just of the radicle we are having hypocotyl. Just above the hypocotyl we are having cotyledon. Just above the cotyledon we are having epicotyl. Just above the epicotyl we are having the plumule. Look at this figure. Here, this is the radicle, this is the hypocotyl, this is the epicotyl. Now this is the cotyledon. Sorry, now this is our plumule. Getting this point? Hello.
Yes, sir. Yes, sir.
Okay. Actually, this small structure that you are seeing inside the groundnut seed, it is like that. Okay. Now, look at, so that here the process of development of embryo is known as embryogenesis. And then look at, and then at the time of the embryo development, the zygote is one cell. So the zygote divides to form apical cell and basal cell. In some plant, what happened? Either the complete embryo develop from the apical cell, or both the cells participate in the development of embryo, or sometimes apical cell contribute a large extent in the development of embryo, but whereas the basal cell contribute the less extent that we have discussed the latter. Look at, and from the zygote, just before the development of the organ of the embryo, the entire structure is known as proembryo. Now come to the development of embryo in the monocot and dicot. The embryo development of the monocot and dicot, they are nearly about the same in the initial stage of the development, but later it varies. And now here we are having the 5 types of embryo development in case of the zygote here, according to the Snarp and Johansson in 1945. Then it has been divided on the basis of then which cell participate in the development of embryo, whether apical cell or basal cell, whether they are dividing the transversely or longitudinally. Look at this here. And then what are the types of embryo? One is the crucifer type. First, what happened? First, this zygote divide to form apical cell and a basal cell. Look at here. The figure A. Figure A is here. The upper one is apical cell. Then the lower one is the what? Basal cell. And here, apical cell A of the 2-cell proembryo divides by longitudinal wall. Remember these things only and only you have to understand that. And there are the following types of the embryo development there. One is the crucifer type, asteroid type, Solan type, Caryophylla type, and the Ctenopodia type. Means and all are the types of embryo that found in the constant family. Crucifer means of cruciferaceae or brassicaceae, and the cruciferous type of the embryo development found in our cabbage, mustard, etc. Astra type, Astraceae. Then Solan type, Solanaceae. Caryophylla type, the family Caryophyllaceae. There's a Crassulaceae, Caryophyllaceae, and then one is the Ctenopodia type. Okay, so that here then, and we can have our clear idea, and when we discuss about embryo development by taking the example. Look at. So now here, at first we've discussed about embryo development in the dicot by taking the plant Capsula bursa pastoris, and then it is the cruciferous type of the embryo development. Look at. So that in case of the plant, look at, and then to study the embryo development in the dicot, we have taken Capsula bursa pastoris as the typical example. Got this point? Hello. Hello.
Yes, sir. Yes, sir.
Look at it. And simply, and if you understand embryo development in our Capsula bursa pastoris, then we can also development. Then also we can also understand how embryo development takes place in the various dicots. So first come to the Capsula bursa pastoris. This is the zygote. First, the zygote divided by transverse wall to form apical cell and basal cell. Then apical cell divided by longitudinal wall to form. And look at, and here the apical cell divided by the longitudinal wall, whereas basal cell divided by transverse wall to form a T-shaped proembryo. Here the two resultant cells that produced due to the transverse division of the basal cell on a CM and CI, and upper cells are two sides. They are present side by side. And then the two upper cells, now they undergo transverse division to form the fourth cell. Now this one is the quadrant stage. First the T shape, then the quadrant stage is formed. Then after what happened? Then each cell of this quadrant or this four celled now become divides by transverse division to form an octant stage. Here CM and CI is there. Then after what happened? Then these the resultant 8 cells which are then and which are here, all the divided by the periclinal division. Due to the periclinal division, we are having two layers are formed. One is the inner cell mass and the outer layer. The outer is known as outer dermatogen. The function of the dermatogen is to give rise to the epidermis, whereas the inner cell mass, which functions produce entire part of the plant, that is the inner side. That is the ground meristem, procambial, the procambial system of the hypocotyledon or the hypocotyl and a cotyledon. Then what happened? Then the cell CM and CI now they undergoes the repeated transverse division. due to the repeated transverse division to form a longitudinal row of 6 to 8 cells. And here what happened? This apical cell start to enlarge in size, and it act as the haustorium. The enter is act as the suspensor. Whereas this the distal cell now becomes start to enlarge in size, and this cell, the pointer where I am pointing, this distal cell now start to enlarge in size, now act as hypophysis. Now this cell undergoes then repeated division to form the two tiers of the fourth cell is. Look, and here this hypophysis undergo division to form the the 8 cells which are arranged in the 4 tiers. Look at now, this is the octant part. Now this is the inner cell mass, this is outer cell mass. Here the two sides of this octant now start to grow out from the cotyledon. Then there is a depression is there, and from this depression, then the pleme will start to develop. Look at here. Now come to the cells that have been produced from the hypophysis. The upper 4 cells that is resultant from the division of the hypophysis, and they gives rise, the 4 distal cells gives us the root cortex, whereas the lower 4 and the proximal group of 4 cells, the root cap and a root epidermis. Look at these 4 cells gives rise to the root cap and a root epidermis. These 4 cells, they are responsible for the formation of the root cortex or the interior portion of the root. And then the two sides, and they are the cotyledon. This part is responsible for formation of hypocotyle. This part is epicotyle. This is the pleural. Got this point? Hello.
Yes, sir. Yes, sir.
Okay. Now this is the embryo development in case of our dicot. So that, and for the dicot we have to take the example of the Capsula bursa pastoris. Got this point? Hello.
Yes, sir.
Yes, sir.
Yes, sir.
And then next come to the embryo development in the monocot. For that we have to take example of Lucilla frosteri. Look at now, this is zygote. In case of the Lucilla frosteri, what happened? The zygote divide to form the 2 cell, apical cell and a basal cell. Now this apical cell and this apical cell, look at this apical cell divided by 1 transverse division by 1 vertical division to form the 4 cell, and then this basal cell undergo 1 transverse division. Look at here to form 2 cell M and Ci. Now this Ci, now this M cell remain constant. This Ci cell become divided to form cell N and cell O. Clear? And here, and all these particular, look at these upper 4 cells. They all undergo the transverse division. Then to form our 8 cell. Now this is the quadrant stage. And then after what happened? Look at. And then after what happened? This particular upper 4 known as the L tier, whereas the lower known as the L tier. Is the cell M, N, look at, and then here this is the O cell now divided by transverse division to form the cell N prime and P. And then from this particular structure, the entire embryo of the monocot become developed. And then L prime that gives us to upper part of the cotyledon, hypocotyle and plumule. The L tier which gives us the basal part of cotyledon, M which gives us the periblem, a part of root cap, N remaining part of root cap, the N prime and P they gives us the suspensor. Suspensor always push the embryo into the endosperm to draw the food material. Okay. Hello.
Yes, sir. Yes, sir.
Okay. Now come to polyembryony. Now tell me, what's you think about the name polyembryony? Then is there any guess? If anyone asks you about polyembryony, then what do you understand? Only you tell me by seeing the name. By seeing the name.
Formation of many embryo.
Poly means what? Many, then embryo.
From a
Yes.
single.
Okay, and simply and when we sow a single seed, then how many plant develops? गोटे मंजी पोतिले हमर केतेटा गछ होउची?
Gote.
गोटे
Gote.
गछ. Why? Because one seed contain one small plant. This is known as embryo. When one seed contain one embryo or one very small plant, when you sow it, then it develop into a single plant. And when a seed contain many embryo or many small immature plant, then when you sow this seed, it become develop into the many seedling or many plant. So occurrence of more than one embryo in a seed is known as polyembryony. Then got this point?
Yes, sir.
Okay, so that here the phenomenon of polyembryony was first reported by Antonio von Leeuwenhoek in the year of 1979, right, in the seeds of orange. Because sometimes when we sow a seed of an orange which produces more than one plant, the Antonio von Leeuwenhoek first time observed this. So question arises, then how more than one embryo present inside a one seed? How do the more than one embryo become developed inside a seed? And then the major causes are cleavage of the cleavage polyembryony. It means suppose we are having seed, the seed having one embryo. If this embryo become divides into two embryo, and then that cause also polyembryony. So that the first cause is the cleavage of the polyembryony or the cleavage polyembryony. Second is the formation of embryos by cells of embryo sac other than the egg. Then third condition is the development of more than one embryo sacs within the same ovule. Development of embryo sac from any sporophytic cell of the ovule. All these are the four causes of the polyembryony. So first you have to discuss the causes. The first is the cleavage polyembryony. Suppose we are having a seed is there. Inside this seed only one embryo present. If due to any cause, if this embryo become fragmented into two or into three, the number of these is equal to number of embryo. Got this point?
Yes,
Hello?
sir.
And then this is the cleavage polyembryony. Look at here. This is the cleavage polyembryony. Here the cleavage occurs. Okay. This is one. This is two. And then the cleavage polyembryony found where in case of the erythronium. This is the cleavage polyembryony. Look at now. Next come to the next cause is the origin of embryo from cells of the embryo sac other than the egg. As you know that egg after fertilization develop into embryo. But sometimes what happen, synergid cell, our the antibodal cell, as well as are the primary endosperm nucleus, they also develop into embryo. So in this case, when the egg become fertilized by the male gamete, it develop into embryo. This embryo is the diploid embryo. But when the antibodal cell develop into embryo become haploid. So in case of plant like allium, piaggio, sedum, almus, here the antibodal cell may develop into embryo in addition to the zygotic embryo. In case of the adjimon and a fasciolus, synergid may develop into an embryo. Okay. In case of the fertilized synergid, in case of Sagittaria, Poa, Aristolochia, Cripis, etc. Got this point?
Yes, sir.
Yes, sir.
Yes, sir.
Next cause is embryo developing from additional embryo sac in an ovule. Now tell me, inside in an ovule, generally how many embryo sac are there? Only one embryo sac, and we say it as the female gametophyte. And when inside an ovule there is more than one embryo sac present, the number of embryo sac is equal to number of egg is equal to number of the embryo, because when egg become fertilized to form the embryo. Getting my point?
Yes, sir.
Yes, sir.
Yes, sir.
Embryo developing from additional embryo sac in an ovule. Then, and for that we are having the three cases. One is by differentiation of more than one megasperm mother cell in an ovule. If there is more than if there is more than one megasperm mother cell, means more than one embryo sac should be there. And by activation of two or more spore, suppose we are having an ovule, this ovule having one, and suppose this ovule having one megasperm mother cell. When one megasperm mother cell undergo meiosis to form the 4 megaspore, if out of the 4 megaspore, then as you know only one survive, the micropellar part survive and the Chalazar 3 become dead. In the last class we had discussed, if out of the 4, if 2 survive and 2 died, then the 2 survive, they develop into embryo sac. So they develop the embryo in the future. And sometimes what happen, from the cells of the nucellus or the embryo sac also develop in the citrus, mangifera, opensia, etc. Sorry. And then in case of this, some cases what happen, in case of citrus, mangifera, opensia, mane amaro otre hela amaro ke citrus, lembu, tapare eta ki mangifera indicate kono, then what is scientific mango. It's amar kon thae ki ei manekar directly some cells of the nucellus or integument, they directly act as zygote and they undergo same pattern that the zygote divide to form embryo. Here there is no embryo sac is formed. And this concept is known as adventive embryos or adventive embryony. The maximum time this question come in the PG entrance examinations. Okay, adventive embryony found in the mangifera, citrus, opuntia, nagopani gacha. Okay. So that to explain our the polyembryony, number of theory have been proposed. One is necrohormone theory, recessive gene theory, and then there is no need to explain about this theory. Then the role of the polyembryony in plant breeding and horticulture. Then in case of simply here the polyembryony helps for the rapid multiplication of plant, because when we sow a single seed, then a single plant develop. But when we sow a single seed, the many plants are developed. So it is possible to rapidly multiply this plant or to rapidly propagate this plant for the agriculture. Then, and whatever the plants which are developed due to the polyembryonic condition, they are more healthier and more better than the normal cutting plants. Okay. Hello.
Yes, sir.
Yes, sir. Yes, sir.
Look at, then the next concept is the apomixis. Look at, and generally as you know that apomixis means what? And then the formation of an embryo without act of fertilization. So that embryo form, but there is no fertilization. So this process is known as the apomixis. Or technically you may say that the phenomenon of substitution of sexual processes by asexual method is known as apomixis. And the plant which show apomixis is called as apomictic plant. Then Winkler 1908, according to the Winkler 1908, the term apomixis refers to substitution of sexual reproduction by any such method which does not involve meiosis and syngamy. Only just remember, the formation of an embryo without act of fertilization is known as apomixis. Embryo means what? মানে ছোট গছ কুন্নি গছ কিংবা ইম্যাচুর প্লান্ট, তাহলে এমব্রিও তৈরি হচ্ছে, প্লান্ট তৈরি হচ্ছে, but there is no gametic fusion. So this concept is known as apomixis. Got this point?
No, sir.
Yes, sir.
Look at, and suppose we are having a female plant, and then and suppose we are having a flower is there. Suppose this flower is unisexual. Unisexual means it having calyx, corolla, and gynoecium. That is a female flower that is nearby your home, and but the male plant of that type is now near your uncle's home. The distance about 10 kilometers, and then the male gamete from that male plant cannot reach to your place because of the high distance. So in case of the male plant, male flower develop having calyx, corolla, and rhizome. the plant that is nearby your home, the female flower develop having callus, corolla, and gynoecium. Inside the gynoecium means what? There is carpel is there. Carpel is consist of stigma, style, and ovary. Inside ovary what present? Ovule present. Inside the ovule our embryo sac inside our egg present. Suppose the male gamete failed to reach the stigma. In this case, what's happened? If the male gamete failed to reach the stigma, in this case, the egg say, I will develop into an embryo because I know that the male gamete will not come. So that in absence of male gamete, I have the capacity to develop into an embryo. Now, in this case, the egg act as like zygote. What it is not zygote because a haploid is egg now behaves as zygote and it follow same pattern of the development as zygote follow for the development of embryo. So to form a small embryo means a very small plant. When we sow this embryo, then it develops into a plant. But this plant is haploid or diploid? This plant is haploid or diploid? Hello. And is this plant is haploid or diploid?
Haploid.
Haploid. Then and here the embryo become developed, but there is no fertilization. So this is not apomixis. And then Moisuri in 1950 recognized the following 3 types of apomixis: non-recurrent, recurrent, and an adventative embryonic or adventative apomixis. First comes the non-recurrent apomixis. If the apomixis embryo develop, if the embryo are haploid, known as non-recurrent apomixis. So the non-recurrent apomixis are 2 types. One is haploid parthenogenesis and haploid apogamy. And those who have and on the audio, please switch off it. And when you have any doubt, you may ask. And while you switch on the mic, look at. As come the haploid parthenogenesis. In case of haploid parthenogenesis, what happened? Look at first our apomixis the 3 types: non-recurrent, recurrent, and adventative. In non-recurrent, embryo develop, all are haploid. It is 2 types: haploid parthenogenesis and haploid apogamy. First come to haploid parthenogenesis. When the egg develop into an embryo without fertilization is known as haploid parthenogenesis. Got this point? Hello?
Yes, sir. Yes, sir. Yes, sir.
And it is found in the solanum nigrum, orchis, then the Cephalanthera. Okay. And then now come to our the haploid apogamy. So that in case of our the embryo sac, where the egg is present here, but and if in embryo sac egg do not develop into embryo, but besides egg, suppose synogyre cell, antipodal cell, they are developed into an embryo known as haploid apogamy. The embryo is haploid. It is found in Lilium, Virginia, Erythrea, okay, etc. Now next is the recurrent apomixis. The recurrent apomixis means whatever embryo or form are the diploid, they are the 2 type: generative apospory and somatic apospory. In case of generative apospory, what happened? Look at and suppose we are having archosporial cell, and I had told you first, and then during our the megasporogenesis, what happened? One cell just below the nuclear epidermis become enlarged in size. It acts as archosporium. This archosporium in the future develop into our megaspore mother cell. This megaspore mother cell undergo meiosis to form the 4 megaspore. Then 3 die and 1 survive. But here is the development of diploid embryos from the diploid cells of the archosporium. When the diploid cells of archosporium develop into an embryo, it's the development of diploid embryos from diploid cells of from the archosporium, and it is commonly found in the Parthenium. And then next is the somatic apospory. It is the development of diploid embryo sac from the cells of the nucellus or integument is known as somatic apospory. In this case, what happened? And besides our megaspore mother cell, another cells of the integument or our the nucellus, and they are also responsible for development of diploid embryo sac. For example, in case of the megaspore mother cell, that the meiosis occur. Then after the meiosis, the 4 megaspore form. Out of the 4 megaspore, 3 died and 1 survived. Thus, these survived, which become developed into the embryo sac, containing 3 antipodal, 1 central cell, 2 synergid cell, and 1 egg. At the same time, one of the integument cell or one of the nuclear cell act as and then act to develop an embryo sac. But the cells are diploid, but here then there is no meiosis occur. And instead of that, this the nucleus become divided by mitotically to form the diploid embryo sac. Hello, got this point?
Yes, sir.
Yes, sir.
And then it is found in the heresium. Next is the adventive embryony. In case of adventive embryony, then there is no any embryo sac form. Only the cells of nucleus or integument that directly act as zygote, and they follow same pattern as the zygote follow for the development of the embryo. That we have already discussed in case of the polyembryony. It is found where in case of citrus and mangifera, citrus, mangifera, eugenia la jamu era देखो mangifera, citrus, boxaceae, orchidaceae, euphorbiaceae, etc. Then this is about apomixis. Next come to our the pollen pistil interaction and self incompatibility. So first come to concept of what is self incompatibility. When the pollen grain of the same flower and do not germinate or fertilized the egg of the same flower, or when the pollen grain of the same plant will not germinate on the stigma and fertilize the female gamete of the same plant, is known as self incompatibility. Got this point?
Yes, sir. Yes.
Hello.
Yes, sir.
Now tell me what is self incompatibility? What is self incompatibility? What is? Now tell me. Anyone?
It prevents inbreeding and promotes outcrossing.
Okay, simply what I haven't told you, just not tell me what is self incompatibility. ଯେତେବେଳେ ସେଇ ଗଛର ଯେତେବେଳେ ଗୋଟିଏ ଫୁଲର ପୋଲେନ୍ ଗ୍ରେନ୍ ସେଇ ଫୁଲର ଷ୍ଟିଗ୍ମା ଉପରେ ପଡୁଛି, ବଡ ଯେ ଜର୍ମିନେଟ୍ ହଉନି କି ପୋଲେନ୍ ଟ୍ୟୁବ୍ ତିଆରି କରୁନି କି ଫର୍ଟିଲାଇଜେସନ ହଉନି, କି ସେଇ ଗଛର ଗୋଟେ ପୋଲେନ୍ ଗ୍ରେନ୍ ସେଇ ଗଛର ଆଉ ଗୋଟେ ଫୁଲ ଷ୍ଟିଗ୍ମା ଉପରେ ପଡୁଛି, କି ସେ ଜର୍ମିନେସନ ହଉନି କି ପୋଲେନ୍ ଟ୍ୟୁବ୍ ତିଆରି ହଉନି କି ଫର୍ଟିଲାଇଜେସନ ହଉନି କି ମଞ୍ଜି ହଉନି, ତାକୁ ଆମେ କହୁଛେ ସେଲ୍ଫ ଇନକମ୍ପାଟିବିଲିଟି। ତା ସେଇ ଗଛର ମେଲ୍ ଗାମେଣ୍ଟ ସେଇ ଗଛ ଫିମେଲ୍ ଗାମେଣ୍ଟ ସେ ଫ୍ୟୁଜ ହଉନି। କ୍ଲିୟର? Hello.
Yes, sir.
And then look at so that here and then simply here and we are having the many causes for that. Then there is our the physiological basis of self incompatibility, genetic basis of the self incompatibility. So first we discuss about the genetic basis of self incompatibility. Look at, and here also then I had told you then we are stigma is always there. The stigma 2 type, one is dry type and wet type. In the dry type, then there is no stigmatic fluid. Wet type, there is just stigmatic fluid present. Look at here. And the incompatibility means, look at here, the in case of incompatibility may be 2 type, intraspecific and intraspecific. Intraspecific means between the 2 species. Intraspecific means the member of the same species. Hello? Incompatibility the 2
Yes,
types.
sir.
Intraspecific and intraspecific. The intraspecific means suppose mango plant and a orange plant. These are the 2 species. So definitely the pollen grain of the one plant do not germinate on stigma of the another. If it is intraspecific means in between mango plant or among the mango plant, among the orange plant. This is the intraspecific. That is the member of the same species known as the intraspecific. So the interspecific may be our heteromorphic, may be the homomorphic. So look at these things. Now come to the genetic basis of incompatibility. And then here to explain the genetic basis of incompatibility, The opposition S allele concept was given by East and Mangelsdorf in 1925. According to East and Mangelsdorf, they कहले कि the allele or gene which present in the pollen grain, if the same is present in our the ovary on the female plant, that will not germinate, that will not able to fertilize, and that cause the incompatibility. Got this point? Hello.
Yes, sir.
Now tell me what is genetic basis? कौन कहले बे? कौन कहले बे? Genetic basis टे कौन कहले? Genetic basis रे केते बळे हमर incompatibility हबो? जेते बळे हमर देखो पोलन ग्रेन छ देखो ए पोलन ग्रेन भितरे जो आलीली थिबो कि जो जीन थिबो यदि सेई जीनटा हम ओभारी भितरे जे रहि जाईची ताले सेई पोलन ग्रेनटा जर्मिनेट करिवो नै जदि करिवो अल्प बटा से रहि जिवो. Look at now come to the first case. Here the ovary contain S1 S2, all pollen grain contain S1 S2. Is there 100% incompatibility or not, or there is a compatibility? Is there a compatibility or non compatibility?
Compatibility.
Compatibility kim dila? Then I have been told you, if the allele or gene which is present in the pollen grain, if they are present inside the ovary, they will not germinate. No, the pollen tube will not form. If form, they are having limited growth, then there is no fertilization occur. Here S1 S2 present in the ovary. Here look at in the pollen grain all having S1 S2. Then will the pollen tube grow? Hello.
Sir.
No,
कौन
sir.
है incompatibility ना compatibility हो?
Incompatibility.
Now come to case B. Now come to case B. Case B is compatible or incompatible? Case B is compatible or incompatible? I speak, look at, and I speak with confidence. If you may wrong, it's okay, no matter, but we'll explain. Speak freely. The case B is compatible or incompatible? What? Look at there. Here, mother contain S1, S3, S1, S3. Pollen can contain S2, S3. Then, what happened? What happened then? Compatible or incompatible? 50% compatible, 50% incompatible. Okay. Then look at it. In case of the C case. In C what happened here? In C what happened? In C what happened? Compatible or incompatible? If compatible, how many percentage? If incompatible, how many percentage?
Compatible.
Compatible.
That's 100% compatible. Okay.
Yes, sir.
Look at, and then about the incompatibility, the two type. One is gametophytic self incompatibility and a sporophytic self incompatibility. And simply here I'm giving some concept of about what is gametophytic self incompatibility, what is sporophytic self incompatibility. Always you think that then we are having pollen grain having outer exine, and then just below the exine the intern is present. So exine is synthesized by the tapetum. The protein present in the exine that is deposited by the tapetum. So tapetum is the tapetum having sporophytic origin. So and then when incompatibility due to the exine protein is known as the sporophytic self incompatibility. Sometimes intern also brings about incompatibility. बेले हमरो exine माने कोन को हम compatibility करंती देखो हम exine पाई तो गजा हुए नहीं कि जर्मिनेशन हुए नहीं हम पोलन ग्रेन ता स्पोरोफाइटिक सेल्फ इनकम्पैटिबिलिटी. When intern helps in the incompatibility known as the gametophytic self incompatibility, because intine is synthesized by the genome of the pollen grain or the gene of the pollen grain. Got this point?
Yes, sir.
Now tell me, then actually what is responsible for the SSI? Exine or intine? Exine or intine?
Exam.
Exine. Then what? Then what are responsible for the GSI? What are responsible for the GSI? Gametophytic self incompatibility. Exine or intine? Hello.
Intine.
Is the intine. Means of protein which was in the intine that is synthesized by the genome of the pollen grain. But whereas the protein found in the exine is synthesized by the tapetum. Now tell me, the exine is chemical composed of? Exine is chemical
Site.
composed of sporopollenin. In the last class I had told you. Look at. Now come to dispersal of fruit and seed. Then before discuss about the dispersal of fruit and seed, first we have to look at the structure of the fruit. Look at. Now this is the structure of the fruit. And here the fruit is developed from the ovary. So the fruit is our matured ovary. What is? Technically the fruit is the matured ovary is known as the fruit. The matured ovule is known as the seed. Okay, got this point?
Yes,
Hello.
sir.
Okay. And suppose we are having brinjal. If we speak, then which is the fruit of the brinjal? Have you eaten brinjal? Have you seen brinjal?
Yes.
ତାହେଲେ ବ୍ରିଞ୍ଜଲ ଫ୍ରୁଟ୍ କୋଉଟା? ପିଲାମାନେ ଦେଖ ସବୁବେଳେ କହୁଛି ନା ମଞ୍ଜିର କଭରିଂଟା ହେଲା ଫ୍ରୁଟ୍. Now tell me, which is the fruit of the brinjal? Which is the fruit of the brinjal?
Brinjal is a fruit. Sir, brinjal is a vegetable or fruit?
Then I'm saying, look at, and here then you are also eating by after the food processing, so that you are saying it as the vegetable. Until brinjal is a first is a fruit, then is a vegetable, and for your the eating purpose. Okay.
Okay, sir. Okay, sir.
Now tell me simply, and then which is the fruit of the brinjal? Brinjal fruit ko part. I haven't told you. The covering of the seed is known as fruit. Then fruit koda? Pericarp. The fruit is technical known as pericarp. তাহলে বাইঙ্গনের ফ্রুট কোটা? কো পার্টটা? কো পার্টটা?
ସେଥିରେ ଯୋଉ ମଞ୍ଜି
ଦେଖ ତମକୁ ଯେତିକି ପଚରା ସେତିକି କହିବ. ବାଇଗଣ ଦେଖିଛ ମଞ୍ଜି ଅଛି, ମଞ୍ଜି ଚାରିପଟେ ଧଳା ଧଳା ଦେଖିଛ? ଯୋଉ ପାଟା ମଞ୍ଜି ଯୋଉ ଜାଗା ଯୋଉ ତମେ ଦେଖିଥିବ ମଞ୍ଜି ଚାରିପଟେ ସବୁ ଯାକ ହ୍ୱାଇଟ୍ ପାର୍ଟ ଗୁଡାକ ଅଛି. ଏ ସବୁ ସେଇଗୁଡା ହେଲା ଫ୍ରୁଟ୍. ମଞ୍ଜି କହିବା ମଞ୍ଜି କଭରିଂଟା ହେଲା ଫ୍ରୁଟ୍. Got this point?
ମାନେ ସେ ବ୍ରିଞ୍ଜଲର ଭିତରେ ଯାହା ଥିବ ସେଟା ଫ୍ରୁଟ୍ ସାର୍.
ମାନେ ମଞ୍ଜି ଚାରିପଟେ ଫ୍ରୁଟ୍.
ବ୍ରିଞ୍ଜଲ
whatever presents around the seed known as fruit.
Okay, sir.
Clear?
Yeah,
The
okay,
fruit
sir.
is technically known as pericarp. Suppose you take example of the mango, and then now tell me, then here, which part of the mango is your fruit? Which part is seed? Look at, in case of the mango, then the upper leathery part is then look at upper leathery part, the fleshy part that you are eating, and then the inner stony part, the takua. What you are saying is whole is known as the fruit. Got this point? ଉପର ଚୋପାଟା ପାଚିଲା ଭାବି ଦିଅ. Look at the right figure. पाचिला आंबर चोपाटा जो पार्ट तुम खाउछ उज टाकुआ ये तुम फ्रूट रो तीनटा लेयर हो गला ऊपर जो चोपाटा खाई की फिंगी दोछ हला एपिकार मेजोकार तुम ढोकी दोछ पेट भीतर को एंडोकार्प टाकुआटा हला एंडोकार्प टाकुआ भितर देखो कोइली अछि कोइली चारिपटे छोटिया गोटे व्हाइट रो कवरिंग अछि दिस व्हाइट कवरिंग इज द सीड कोट तो कोइली itself is the seed कोइली with this covering the seed कोइलटा ताला गोटे कटलीडन व्हाट इज पॉइंट
Yes, sir.
दीटा देखो कोइली दीटा कोइली कोइली भितर छोटिया स्ट्रक्चरटा अछि एम्ब्रियो ता दीटा कोइली ला कटलीडन ता भितर एम्ब्रियो अछि ना एम्ब्रियो कटलीडन आर द पार्ट ऑफ द एम्ब्रियो आल्सो ताले पूरा कोन ये हमर मंजी हे गेला माने कोन देखो कोइली प्लस एम्ब्रियो रहिला एमिति चारि पोट धळा थिबो ओटे कवरिंग आंब काटला सब देखिबो
हाँ सर.
ते कवरिंग मिसी की सीड हे गेला ताले फ्रूट के अरे फ्रूट के
से जो तार लेयर ता ऊपर थिक लेयर टा प्लस से भीतर
फास्ट टाकुआ
हाँ
एंडोकार्प then mesocarp then
मेजर
epicarp
एपिक
then epicarp endocarp mesocarp is collectively called as the pericarp and then the look at here this type of the mango is a type of fruit known as a drupe cucumber pepo cucurbita pepo apple look at the central part you are seeing here apple now this is the fruit but the peripheral part you are eating this is the fleshy thalamus but getting my point the part of apple you are eating they are the thalamus but the fruit is this part this portion is the fruit the black is seed just around the black the fruit is present the remaining part the fleshy part you are eating that's thalamus means what थैलामस माने जहा ऊपर तुम कैलिक्स कोरोला एंडोसोम गाइनोसोम हेइथो डेवलप हेइथिबो द स्टेज ऑन व्हिच कैलिक्स कोरोला एंडोसोम गाइनोसोम डेवलप नोन एज थैलामस लुक एट दिस इज द ना पोमीग्रेनेट दिस इज द ना बालास्टर टाइप ऑफ फ्रूट हेस्पेरिडियम ऑरेंज okay now simply here now tell me in case of coconut in case of the coconut then now tell me then which are epicarp mesocarp and endocarp coconut रे
ବାହାରଟା ଏପିକାର ପୁରା ବାହାରଟା
epicarp then
ତାର ଅଂଶ ମେଜୁକାର
କତ୍ତାଟା ହେଲା mesocarp ସଡେଇଟା ହେଲା endocarp
एंडोक
clear
yes sir
look at the figure here the structure that you are seeing this is the structure of the apple the middle part is known as look at the seed at the center this part is our the fruit the rest of part we are eating this is the thalamus mane the structure or stage on which the different parts of the flower develops okay hello
yes sir yes sir yes sir
ସାର୍ କୁକୁମ୍ବର ର କୋଉଟା ହବ
हां
କୁକୁମ୍ବର ର କୋଉଟା ମେଜୋକାର୍ପ ଏପିକାର୍ପ ହବ
अंता जो सोचा look at here now we are having look at now we are having the dispersal of fruit and seeds means how the seed and fruits are moved or transmitted from one place to another place there's some agency and then wind water animals and some plant also having explosive mechanism for the dispersal look at here and then so that the now first mechanism is the dispersal by wind and then those seeds who are dispersal by wind they're having the parachute mechanism look at they are having hairs and then So that here, and by the means of hairs, they can easily, and then they can also easily travel from one place to the another place. Look at the hairs. Some are having hairs, some are having persistent style. Look at it. These are the balloon-like appendages, rolling mechanism, as the dispersed by water, hydrochloric. Look at, and all these are the examples. And then some seeds are having hook. They also, and then they can also move from one place to another place by attach with the animals. This is known as the, look at, dispersed by animals known as zoochory. Zoochory means what? And then when the food or seed develop move from one place to another place by the help of the animals. ପିଲାମାନେ ସବୁ ଦେଖିଛ ତମେ ଜଟ ଜଟିଆ ଦେଖିଛ? ଜଟ ଜଟିଆ ଗୁଗୁଚିଆ ଜାଣିଛ? ହ୍ୟାଲୋ
ସାର୍
Yes,
କଣ?
sir.
ଆରେ ଗୁଗୁଚିଆ ତମେ ସବୁ ଜାଣିଛ ସବୁ ଯାକ ସ୍କୁଲ ପଢିଲା ବେଳେ ସବୁ ମାନେ ଖେଳ ପଡିଆରେ ଫୁଟବଲ ଖେଳ ହୁଏ, ଆମେ ଯେତେବେଳେ ସବୁ ସ୍କୁଲରେ ଖେଳୁ ହାଫ ପ୍ୟାଣ୍ଟ ସବୁ ପଶିଯାଏ ନା ଗୁଗୁଚିଆ।
ହଁ ସାର୍ ପ୍ୟାଣ୍ଟରେ ଲାଗିଯାଏ। ହଁ ସାର୍, ହଁ ସାର୍
ସିଏ ହେଲା କଣ?
ଦେଖିଛି।
ସିଏ ହେଲା ଆମ ଜୁକୋରି, ବୁଝିପାରୁଛ ତ?
ହଁ ସାର୍।
ଆଉ ଯୋଉ ଗୋଟେ ଜଟ ଜଟିଆ କହନ୍ତି, ଜଟ ଜଟିଆ ଯୋଉ ଛୁଆ ମାନେ ଯୋଉ ମାନେ ମୁଣ୍ଡରେ ପକେଇ ଘଷି ଦିଅନ୍ତି ସ୍କୁଲରେ ପାଠ ପଢିଲା ବେଳେ, ମନେ ଅଛି ପିଲାମାନେ?
Yes, sir.
Yes, sir.
ହଁ, ତାପରେ ଗୋଟେ sticky fruit, and when the fruit becomes sticky ହେଇଥିବ, ଚିପଚିପି ତ ଲାଗିଯିବ, Loranthus, Plumbagos, Cleum, then edible fruit, and then in case of the some plant they have been edible fruit, and then those fruits are edible or seeds are edible, the animals come and eat, and they also transmit them. Dispersed by explosive mechanism or autogory. In case of some plant, what happen? The fruits, the fruits that explode, and when the fruit explode or crack by explosive mechanism, that cause the dispersal of seed. କିଛି ଫଳ ଅଛି ଜୋର ଠୋକିନି ଫାଟିବ, ଫାଟିଲେ କଣ ହବ? ମଞ୍ଜିଗୁରା ସବୁ ଛିଟିକିକି ପଡିବ। ଏକ୍ସାମ୍ପଲ କିଏ ଆସିଲା? ଆମ ଅକ୍ଜାଲିସ୍ ଆସିଲା, ରିସିନସ୍, ଯୋଉ ଗବ ଦେଖୁଛ ହେଲା, ବାହୁନିଆ, ଫ୍ଲୋକ୍ସ, ଉରୁଲିଆ, ଇମ୍ପାଟିଏନ୍ସ, ଯୋଉ ବ୍ଲାଜମ ପ୍ଲାଣ୍ଟ ଦେଖିଛ ଛୁଆଟା? ବ୍ଲାଜମ ପ୍ଲାଣ୍ଟ ଦେଖିଛ? ଯୋଉ ଟିକେ ଟିକେ ତା କାକୁଡି ଭଳିଆ ଗଛ ହେଇଥିବ, ଆମେ ତାକୁ ଗୋଟେ ଛୋଟ ଛୁଆ ବେଳେ ତାକୁ ଗୋଟେ ପତ୍ର ଉପରେ କି ଛେପ ପକେଇ ତା ମାନେ ଫଳଟା ରଖିଦେବୁ, ସେ ଠୋକିନି ଫାଟେ। କରିଛ କେବେ ଏଇଟା? ଇମ୍ପାଟିଏନ୍ସ ବ୍ଲାଜମ ପ୍ଲାଣ୍ଟ। ହ୍ୟାଲୋ
No, sir.
Okay, and if you are not done, it's okay. And then simply you have to remember this is Oxalis, wood sorrel, Ambiradi gacha, Ricinus, Phlox. In this plant, we will find the explosive mechanism. And then these are the seed dispersal mechanism. Dispersed by wind, dispersed by water, by animal, by explosive mechanism by itself. Hello?
Sir.
देखो आउट स्टार्ट करिया बे स्ट्रक्चर ऑफ द सीड खाली देखते पड़िए ना कम द स्ट्रक्चर ऑफ द सीड look at this is a seed here and then the seed having seed coat is known as testa. Okay, and then the some seeds having the outer seed coat known testa. Just with testa we have the tegmen is there, and then this part is the cotyledon, this part is the embryo, this is epicotyle, this is radicle, this part is the hypocotyle. Okay, this is the structure of seeds. Seed coat, embryo, food storage. Okay, these are seeds. Now tell me, and whatever we have studied, is there any problem to understand? Whatever we have studied, is there any problem to understand? Hello. Now tell me, then is there any doubt to understand?
No doubts.
Now give me some example of the endospermic seed. Now give me some example of endospermic seed. The seed having endosperm. And you all knows it. Now give me some example of the some endospermic seed, some non-endospermic seed. Nanjo,
Coconut, groundnut.
especially monocot seeds are endospermic seed. And then the rice we are eating, then it is endosperm. Maize, endosperm. Then we are eating endosperm. But in case of our the groundnut, especially the dicot are the non-endospermic or X albuminous. The dicot what happens? Suppose take example of the groundnut, badam nature. Badam देखो दी फाड़ा होई जे दी फाड़ाटा जाके होला हमर कोटलीडन ता छोटे जो एम्ब्रियो थाए the small depression present in the cotyledon is the place where endosperm had stored but embryo consumed whole the endosperm. So in case of the monocot, then there is the storage organ is the endosperm, where in the dicot the storage organ is the cotyledon. Got this point? Generally, the dicot seeds are non-endospermic and the monocot seeds are endospermic. And then we are having, look at, and then in case of the maize, sometimes we encounter mosaic endosperm. Mosaic endosperm means suppose we are having a maize, ଆମର ଗୋଟେ ମକା ପୁଟ ଅଛି ହଳଦିଆଟେ, this a part become white, a part become yellow. Okay, so that when there is not homogeneous composition of the endosperm, this type of endosperm is known as the mosaic endosperm. Got this point? Hello. Hello. Got this point? Yes or no? Hello.
Yes, sir.
Look at this figure I'm seeing here. Then this is the example of the mosaic endosperm. Look at, and then a part of the seed is white, a part of seed is yellow. This is known as the mosaic endosperm. And then the maize, in case of the maize, and then endosperm is our the storage tissue. But here a part is white, a part is yellow. When the endosperm is not uniform or it is a composite, it's known as the mosaic endosperm. Now next is our the ruminant endosperm. When endosperm having not smooth boundary, but here look at here and this having smooth boundary. When endosperm having not smooth boundary and this known as or smooth outline known as the ruminant endosperm. Now here I am showing the structure of the ruminant endosperm. Look at here. Ruminant endosperm. Look at here. Look at this figure here. And here look at. And in case of here, this is look at the boundary or the periphery of endosperm is not or uniform. Instead, there is some cutting area here. There is some the irregularity are there. So this is known as the what? The ruminant endosperm. Ruminant endosperm is found in Rubiaceae, Annonacea, Palamaceae, Palami, Tala family. Clear?
Passion fruit.
Ruminant endosperm found in our Annonacea, Rubiaceae, Aristolochiaceae, Palmaceae, Maristiceae. In the following, we found the ruminant endosperm. Ruminant endosperm means when the boundary of endosperm is not even. Look at this is our the এটা গুয়ার দেখো ছুয়া এটা হচ্ছে গুয়া দেখুছো গুয়াটা কোন পুরা পুরা গোল হওয়া দরকার দেখো এটা হচ্ছে ফটাফট দেখুছো না ফটাফট সব দাগ আছে দেখুছো না এটি হ্যালো
Yes, sir.
Yes, sir.
দেখো না গুয়াটা কোন আমরা না গুয়াটা কোন দিছে দি না পটলি ডন্ডা না সরি দেন দিস আর দি এন্ডোস্পার্মটা এটা দেখো এই সবটা কটিং আছে দেখো এটা তো কটিং আছে যে পুরা আমরা ইউনিফর্ম নাই এটা আমরা তার কোন তার তো না পেরিফেরি আমরা এটা ইউনিফর্ম নাই so it is known as what our the ruminant endosperm. But in case of our the maize is the example of the mosaic endosperm because the boundary is smooth, but a part of the maize seed is yellow and a part is white. Got this point? Hello.
Yes, sir.
And for your remember and for your kind information, just I'm saying that then your course is vast. The paper that I'm dealing with, a single question can take about 1 or more hours, and only the 4 classes are allotted to me. The 3 classes have been completed, and another 1 class will be day after tomorrow. That is at the 16th. And then I have to complete the whole paper of your the anatomy and our the embryology in the 4 classes. Then you can understand it is not possible, so that I'm skipping then one by one. Actually, if there is more time, then we have to discuss this sequentially. But it is not possible at all. So that and that's it. Just I'm speaking like that. So okay.
Yes, sir.
Now tell me, is there any problem for today? Is there any problem to understand for today?
No, sir.
No, sir.
No.
Okay, and then coming class will be at 16. Okay.
Yes, sir.
Okay.
Thank you,
Now,
sir.
Dr.
Thank